Hawai, tēnā tātou katoa mai, te rorohiko I tēnei whare, ka whakamahana I a koutou, tēnā tātou. Ki a koutou ko ngā kaiwhiwhi mā tauranga, kia kaha I tō hui-huinga tanga I tēnei rā. Kia inoi tātou, whakataka te hau ki te uru, whakataka te hau ki te tonga, kia mākina ki uta, kia mātaratara ki tai. E hiake ana, he atakura, he tio, he huka, he hauhu, ti hei mauriora. As I just said earlier to all of you on the computer in the room in this nice warm whare, welcome. And to our summer scholars, give it heaps, go hard and do your best. Best of luck.
Awesome. So just before I throw it over to our host for today, just a couple of housekeeping things. This Zoom is going to be recorded, and so we can share this onto our peers and fellows after this. So while we are recording this, we just ask that people who are online, if you could just keep yourselves muted during the presentations. And if you've got any questions or feedback for our presenters, feel free to pop it in the chat. And then if you guys are in the room, you can just flag out to me and I'll let Margaret know online that we've got a question from the floor in person. At the end of every presentation is about 5 minutes long. We may have 1 to 2 minutes for questions or feedback, and maybe we'll have one online and one from the floor in person if there's anything here. And if you're presenting online, when it's your turn, you can share your screen. And if you're presenting here, I'll get you guys to sit in the driver's seat so you can navigate your presentation with the mouse, or if you can trust me, I don't mind doing it too. It's up to you guys. I would like to now throw over to our host. Our host is joining us today from the University of Auckland. Professor Margaret Stanley is an oncologist with expertise in biodiversity, biosecurity, conservation, invasive species, urban ecology, and just awesome. She's a researcher of PO 3.2, looking at the impacts of co-occurring weeds with our postdoc, Diana. Please take us away, Margaret.
Tēnā koutou katoa. Welcome everybody. It's really nice to see everybody here supporting our summer students today. I know that some of you will be extremely nervous, but it's great to have such a supportive environment. So looking forward to hearing what's been going on over the last summer. Just from what I've heard, there's been, a few fun and games, but some really good outcomes and some I think really great experiences for the students in terms of what research can be like, and hopefully, as we can see from some of last year's ones, going into masters or honours after that. So great stuff. We'll get straight underway with Bella Perham from the University of Auckland, who's online. Maybe you're ready to share.
Sorry, Margaret, before we do, we actually have a little video to share with the group that we've pre-prepared. So I will just take over for a quick second and share our screen to play our video.
Are you listening? Damn.
Yeah, definitely. 100% science is fun.
I think it is, yeah.
Yeah.
People generally see the outcome of science. They see this nicely put together research, but in fact there's a lot more going on behind the scenes.
A lot of people kind of think that science is about answering questions, and it is, but it's also just as much about working out what questions need to be asked.
I have to say the field, I like being outdoors. Working in wetland areas is quite kind of my jam.
I definitely prefer field work. For me, it encapsulates so much of what I love about biology.
The main reason why I'm studying science, to be honest, because I enjoy those out in the field roles, and you get a very interesting perspective on those places at the same time.
I've actually wondered about that and why bioprotection is named bioprotection.
Research, collaboration,
maybe innovation,
protecting our environment,
Indigenous, and Modi.
I hope so.
I think it will.
That's the very high priority for me.
Any work in biosecurity conservation is increasingly becoming more important.
The climate of today, I think we are going to see a lot of unprecedented change, and I would love to be working at the forefront of that.
I definitely think that in the future what I'm going to be doing will have meaning and importance.
There we go, Margaret.
Well, that was awesome. Glad I didn't stuff that up altogether and we actually all got to see that amazing video. And hear the enthusiasm. If that was filmed after summer, then we didn't turn everybody off completely and we've all left with some enthusiasm, which is fantastic. Okay, so now we're going to throw over to Bella Perham, who's from the University of Auckland.
Okay, cool. Thank you. I'll just share my screen. Okay, can everybody see that all right? Cool. All right, so kia ora everyone. My name is Bella and I had the pleasure of working with Margaret, Diana and Ella this summer in Auckland. And my research asked the question, how does distance to nearest native seed source and surrounding land cover type influence species richness in terms of weed seedlings? So invasive plant species pose one of the greatest threats to Aotearoa's native biodiversity. Due to their prolific growth, they can outcompete native species and alter the structure and composition of ecosystems. Land use type and distance to nearby seed sources are key determinants in their establishment, where the highest levels of invasion are often among agricultural, urban, and industrial land cover types. So weed species richness is simply the number of weed species. And I investigated first the relationship between the proportion of indigenous forest, the surrounding indigenous forest, and weed species richness, and second, the distance to the nearest native seed source and weed species richness. To do this, we surveyed seedling plots with a 1.13 metre radius, which I've done a little drawing on the right. The green circle in the centre is one of the 3 target weed species we were looking at, and that was either woolly nightshade, tree privet, or brush wattle. The surrounding coloured stars are the seedlings, where the blue ones are native and all the other colours are weeds. So here we can see that there's 5 different coloured weed species, so this is what we recorded. Other confounding variables such as slope, litter depth, canopy cover, composition of ground cover, evidence of disturbance or erosion were also recorded, but I didn't analyse any of these. I then went away and used Google Earth and geomaps to approximately estimate the proportion of surrounding indigenous forest within a 50-metre circular radii, as well as distance to the nearest native seed source. So now onto the results. We have 2 graphs. The graph on the left shows the proportion of surrounding indigenous forests on the x-axis and weed species richness on the y-axis. According to my R-square value of 0.2116, we can see that there's a weak relationship between these 2 variables, where there are more weed species interacting when there was less forest. On the right, we can see that distance to the nearest seed source was on the x-axis and weed species richness was on the y-axis. Again, this relationship is quite weak, but we can see more weed species interacting when we're further away from a native seed source. So although these patterns are not as convincing as we would have hoped, they do make sense and they're sufficient in explaining the importance of propagule pressure in weed seedling establishment, especially land type, as this influences what propagules or seeds are available. So if there's less indigenous forest closer and more of other land types such as urban, industrial or agricultural, then there is a greater chance of weed propagule spreading and establishing. And often these ones are garden escapees from people's gardens. So perhaps these relationships were not as strong because of other determinants, such as property owner attitudes. I think at least 4 of the properties we went to, they were already removing and controlling their weed species, as well as the environmental characteristics that we did measure but did not analyse. Also the height of the central species and the size and the location of the sites. Maybe we could also, instead of looking at Indigenous forests, we could look at the other land types. So I'd just like to finish off by saying a huge thank you to Bioprotection Aotearoa for having me this summer. It's been a very special opportunity. It's allowed me to work with passionate and like-minded people, explore parts of Aotearoa, and expand my understanding of invasion ecology. So thank you to you all. That's it from me. Are there any questions?
Well done, Bella. Does anybody have any questions for Bella, either on the floor in Lincoln there or on the chat?
We've got a question here from the floor in Lincoln.
I was just wondering, how did you define what a weed was?
I based it off a document that I found, I think it might have been Auckland Council, and it has all the species, whether they're a weed or introduced, and then all the other ones they were native. So yeah, that's how I did that. Thank you.
Any more questions there from Zoom or on the floor? No? Okay. We shall move on then. Thank you, Bella. Okay, so next we have Haig Bishop from the University of Canterbury, who's hopefully in person there with you, Zaha.
Yes, he is. I'm just going to share our screen and let's back and start. It's yours.
Okay.
Perfect. Whenever you're ready.
Okay. Oh, let me see. Okay. All right. Hello everyone, my name is Haig. Over summer I worked with Sophie and Claudia. Sophie is working on Phytophthora effector proteins. So Phytophthora is a genus of oomycetes which infect plants. And oomycetes are a group of organisms which are similar to fungi but phylogenetically distinct. So the species that we working on is Phytophthora pluvialis, which mostly infects pine species, and so it's a huge, increasingly large issue in the forestry industry in New Zealand. So effector proteins, which I'm not sure if you can see, effector proteins are proteins produced by a pathogen, and they interact with the host to facilitate infection. So one type of effectors is called RXLR effectors, which are found in Phytophthora species and other closely related species. Pluvialis has about 115 of these RXLRs in its genome. And so we narrowed it down to 9 for us to study. So they can be seen down here, PPR 01 through 09. The typical RXLR effector looks a lot like this. It has a signal peptide domain and a translocation domain, which are cleaved upon secretion from the pathogen, and then as it enters into the host. And that leaves you with the effector domain, which is the part that does the interacting. So for instance, it might target a host protein, which might prevent an immune response, which would facilitate further infection. So this is an overview of my practical work. So we use the multiple molecular techniques to determine two main pieces of information for each of the 9 effectors. The first of which is the localization. So that is where in the host cell are these proteins localizing. And the second piece of information is the interaction information. So specifically what in the host cell is being targeted by the effectors. And I also did a bunch of digital work. So this is an overview of my work on the third of those 9 effectors, PPR03. I generated predicted models of all of the proteins. So this is the 3D model, predicted 3D structure of PPR03. I also searched for similar proteins in other Phytophthora species, and I used those those lists of similar proteins to make sequence similarity networks. So here in the top right, we can see a network of other RXLR effectors in a bunch of different Phytophthora species that are all similar to PPR03, similar in sequence. And this can show us how they're clustered together and which ones are similar. And because these sequences are all similar, these proteins are all similar, and they're all in closely related species, it would make sense for us to predict how they are related. So here we see a predicted phylogenetic tree for these proteins. And then here, these colorful bands show a structural analysis of these proteins. So they're similar, but they're not exactly the same. So what about them is generating this similarity, and which parts are different? All of this information can be used to understand the infection mechanism and would help us prevent disease caused by Phytophthora species. So I'd like to thank Bioprotection for funding this research. Thank you to Claudia, thank you to Sophie, and thank you, UC.
Awesome. Thank you, Haig. Has anyone got any questions?
Yep.
What did you use to predict the RXLR effector protein structures? You use AlphaFold, was it like a...
Yeah, so we use, for most of the predictions, we use ColabFold, which is it's a free service, and we also use RobettaFold for some of them. So I did models for as I said at the start, there was different cleavage points. So we predicted structures for the full protein, and then the protein after each cleavage. Yeah. So ColabFold. That's the answer.
Did you like it? Was it useful? Is it easy to use?
Oh, it's not very pretty, but it works. Yeah, and it's not fast either, but yeah.
Any other questions for Haig? I have a question. Is that? Oh, sorry. Carry on.
You're good to go.
No, I was just going to ask if this was something you were keen to carry on into a master's or honours, or put you on the spot there.
I'd love to keep on going. This is Sophie's project, so I think I'm going to watch from the sidelines and keep updated with how Sophie's going with this. I'm interested to see where it goes,
Awesome. Thank you. Okay, so next up we have Zelia Swartua, who's beaming into us from my hometown of in Dunedin, from Otago Uni. So a very warm welcome to you. Not biased at all. And we will get you sharing your screen and showing us what you've been doing.
Cool. I'll just get that going. Cool. Can everyone see that screen? Yeah. Hi everyone. My name is Alia. I'm from Dunedin, studying at the University of Otago. My summer project was looking at identifying symptoms of early colony decline in honeybees using prognostic tools of health with Peter Dearden's lab, with one of his postdocs being my primary supervisor. So just a brief intro onto kind of what I was mainly looking at. I was looking at the varroa mite, which is one, as you can see in like the blue circle, it's an ectoparasitic mite which attaches to honeybees of all stages of their developmental life, and basically it feeds off their fat bodies, which kind of disrupts their immune system and such. And it also is a vector that transmits the virus, deformed wing virus, which is the most prominent pathogen in honeybees, resulting in crumpled wings, as you can see, and also shortened abdomens. So why this project is important, I think it's, well, honeybees they enhance our genetic diversity in crop species and such. They also increase the yield, quality of fruits and seeds, and their overall health of the hive is kind of an indication of the environment that they're surrounding in, which is quite important to us. So the overall aim for this project was to see if we can identify those early signs and to create possible preventative measures further down the line. So this is just a kind of brief-ish summary of what my summer project looked like. So we were lucky enough to go up to the Midland apiaries up in Ashburton, where we collected samples from 50 hives, which we then used to analyze and get our data from. So as on the two images on the left side of the screen just shows, one is a normal and then another is the kind of most severe affected honeybee, which you can see indicated by the arrows, shows really crumpled up wings, which compared to the nice flat laid out wings in normal ones. So this graph just kind of shows the influence from the varroa mite on the deformed wing severity score, we kind of called it, on a scale of 0 to 5, 0 being it wasn't affected, 5 it was highly affected, which is, as you can see by this, which both wings were real kind of thread-like. So we found that when there was more than 10 varroa mites present in the colony, there was a kind of more prominent number of honeybees that came out with a higher severity score, with higher score of deformed wing. And we also found from the RNA concentration of the deformed wing virus, obviously a higher concentration of copies found with the higher varroa mite. And this was just a quick, because we collected data from August of last year till December, and there's a clear kind of real quick drop in varroa mite numbers, which was quite interesting to see because we thought there was a big problem in those apiaries, but it's good that their numbers are dying down. So, and so this was looking another expression levels of vitellogenin, which is a protein involved in the immune system of the honeybee, and that's produced by the fat body tissue, which we know that the varroa mite feeds on. So when varroa mite were present in the colonies, we found the expression level of this vitellogenin to have decreased compared to when they were absent from the hives, which means, the varroa mite are kind of affecting their immune system. The honeybees themselves aren't really surviving to later stages. So this was just a kind of quick conclusion of my overall findings and stuff. So we found when the colonies have a greater mite infestation, there's a higher titer of deformed wing virus, which we found result in a higher number of honeybees emerging with deformed wings. And yeah, this deformity is scalable from, we've found different sort of descriptions for each scale of deformity. So like scale 5, meaning they're like both wings are thread-like, as you saw in the previous images, and then kind of going down, it gets less severe, if that makes sense. But yeah, and we also did some vibroacoustic collection data, which we basically recorded the sounds that the bees were making, that haven't really gotten to look at that data, still under review at the moment, but hopefully I'll be able to include that in my final report and such, yeah. And yeah, and just a quick thank you to all my supervisor, Carol and Peter Dearden, and also everyone in his lab that helped me throughout my summer project. It was good time, fun times. And also Bioprotection Aotearoa for their involvement in making this project possible and so thank you.
Awesome. Thank you so much, Salia. Anyone got any questions? Yes. Katerina.
Maybe I had to type it right.
Can you repeat that?
Yeah, can you hear me?
Not really. We can't hear you very clearly. Did someone on site while Katerina is typing that have a question down in Lincoln?
Yeah, I had a question. It's Nicola Sullivan here. Nice talk, Zalia. That was really good. Did you ask the APRIS about their treatment regime, like when they treated, because most of the APRIS need to be treated quite regularly for all that?
Yeah, so we found out like a bit later that they had been treating them.
That's usually the way.
Yeah, but it was quite interesting to see the like real quick drop though. Like usually it's quite a progressive decline, but this one was like high and then just completely drops almost zero in like 2 months, which we found quite interesting in a way, but they might just be really good at getting rid of them. So that's also another.
It would be interesting to know what treatment they use as well.
Yeah, I'm not sure exactly what kind of treatment they did, but yeah, all we know is that they were carrying out the treatment and so.
Awesome. Yeah.
Great. Katerina's written in the chat here that she was interested in the distribution of the virus. Is it widespread in New Zealand or is it localized at the moment?
I think I'm not sure about right throughout New Zealand, but and it's kind of spread it's a common virus that's like found even when Varroa mite isn't found, like it can just be present in the colonies without actually causing too much sort of prominent symptoms, as we saw like with the deformed wings and stuff. But it's found that when Varroa is present, that those symptoms come out a lot more. They become more prominent. So and a lot of the literature studies and such have shown that like the honeybees, they can survive with quite high levels of the deformed wing virus, but then once the Varroa mite becomes established in it, that's when they start showing the deformed wing symptoms.
Great, thank you. And I have one last question. Did you manage to avoid getting stung?
Yes, luckily.
Well done, you.
Yeah, just from
that's a win.
Yeah, one time it was an interesting having to like go towards the sounds of the bees instead of away from them. So that was quite
counterintuitive.
Yeah, it was a really good experience. I was thankful to be able to do that.
Awesome, thank you very much, and we are pleased there weren't any major health and safety events. Okay, so I think now we have a pre-recorded talk from Oscar Ballot from the University of Canterbury that hopefully Zohar will share with us in a second.
Yes. One moment, just set it up. Just sharing a screen. Share video. And pushing play.
I'm Oscar Ballett, and for my summer project, I looked at whether invasive pines affect fungi and grass roots. I was working under the supervision of Ian Dickey and Joanna Green here at the University of Canterbury. Currently estimated to cover around 1.8 million hectares in New Zealand, wildland pines have a number of adverse effects on afflicted areas. Past research has found decreased nutrient, water, and sunlight availability in these areas, contributing to a decrease in species diversity, ultimately changing the ecosystem. I wanted to look at how this change would impact fungal communities, specifically arbuscular mycorrhizal communities. Arbuscular mycorrhizal fungi, or AMF for short, refers to any fungi belonging to phylum Glomeromycota that form a symbiotic relationship with plants. Put simply, this relationship occurs when a hyphal extension, such as you see here, penetrates a plant root, providing the plant with organic phosphorus in exchange for sugar and lipids. This exchange typically takes place at a site of nutrient exchange, such as coiled hyphae or an arbuscule, which is this tree-like structure you can see here. Given this mutualistic relationship, AMF could be considered an indication of soil or ecosystem health. For my research, I wanted to compare plant roots from areas where wildling pines had invaded to areas of pine-free grassland. Roots examined came from 4 different grass species collected along the Clarence River Valley in Marlborough. 2 of these were native, while the other 2 were exotic. To measure levels of AMF in roots, I had to stain them. So standard staining procedure basically involves stripping the color out of the root and then adding a dye. In theory, this should highlight fungal structures while clearing the root. However, I found this technique wasn't really optimized for New Zealand grasses, and the images it was producing weren't very clear. So after a bit of trial and error, I ultimately ended up finding a more suitable process. And this was from a paper published in 1970. And this drastically improved the quality of images I collected. So once I honed in on this process, I was getting images like the ones you see on the screen here. I spent about 250 hours behind a microscope recording the presence of AMF structures and scanning for various other structures as well. So this gave me the opportunity to record other fungal structures that weren't related to arbuscular mycorrhizal fungi. So we can see on this slide here, we have an oomycete sporulating structure here and some septate hyphae, which are unrelated to AMF. These structures are actually an equally valuable measure of soil health, as while not always the case, they're typically parasites. Once I'd collected the data for this, I was able to test for a relationship between environment, so whether that's pine or grassland, and the levels of AMF and these non-AMF structures here. I also wanted to see if there was a relationship between the individual species and levels of AMF and non-AMF. So still a bit of work to do on the data analysis, but my initial results appear to be really promising. I found a significant difference in the percentage of AMF between grassland and pine areas, with a 3% reduction in average AMF levels in pine invaded areas. There was also a significant difference between the individual species and the levels of AMF. For the non-AMF structures, there was not a significant difference between environment or species, but I was still really happy with the results I got from these AMF recordings. So as mentioned, there's still a bit of work to be done on this, but my data appears to indicate that areas invaded by pine are potentially being affected by that pine invasion, which is decreasing the numbers of that AMF. This aligns with my initial expectations because we already know that AMF are having this adverse effect on areas, so it makes sense that it would affect the beneficial fungi as well. I was really happy with these results, but I guess as the old saying goes, sometimes the real results are the fungal structures you meet along the way. And throughout the project, I came across over 80 different unique structures that I was unable to identify or unable to fully identify. We can see in this slide here, got this parasite that's causing a kind of bulging at the surface of the root here. They've also got this really unique, densely packed sort of cellular structure. Now, aside from being really cool, these, I guess, also serve to illustrate just how little we do know about what's going on with endophytic fungi, and it definitely leaves a lot of room for future research. So that's pretty much it from me. I want to say a big thank you to Ian and Joanna for all the support that they've offered me throughout the project. And if you have any questions, feel free to get in touch at the email on the slide here.
Awesome.
So we can thank Oscar in his absence, and if you're super keen, you can send him an email and ask more about those fungal structures. So now we have a presentation from Lyadon Dickey, who is at Lincoln University and should be in the room there in person, I think.
All right, I'll just set up her presentation. Going to come closer to the mic.
So I spent my summer exploring several methods to detect Phytophthora with the aim of investigating how land use affects abundance and diversity of these critically important plant pathogens. So the first method of Phytophthora detection that I investigated was QPCR. So QPCR involves extracting DNA from the sample and then amplifying a specific target region of that DNA. The product of that amplification is tracked as it accumulates, and you can compare that to a standard curve constructed from DNA of known concentration to work out the initial abundance of that target DNA region in a sample. So I extracted DNA from soil samples from 3 contrasting land uses in Kaituna Valley on the Max Peninsula, and I amplified a region of that DNA that's specific to the Phytophthora genus. So with this method, I found that there was more Phytophthora in soils from exotic forest than there was in soils from irrigated pasture or regenerating bush. So using the standard curves that your qPCR reaction generates, you can assess the quality of your results. Ideally, this efficiency value here should be between 90% and 110%, and the R-squared value should be above 0.99. As you can see, those criteria weren't met with my qPCR reaction. They're not too far off, but it does highlight that you can get poor quality results if you've got imprecise methods, and that can render your conclusions meaningless or misleading. There's also other limitations to qPCR. For example, certain soil components can inhibit the PCR reaction, and certain life stages of Phytophthora are harder to extract DNA from. Additionally, you need that DNA that you construct your standard curve out of to amplify with the same efficiency as your target DNA. And if you're targeting DNA from multiple species, like the whole Phytophthora genus, that might not be possible. So the second method of Phytophthora detection that I investigated was soil dating. So soil dating involves a series of steps that encourage any Phytophthora present in your soil sample to produce sporangia. You then flood your soil sample with water, And those sporangia release zoospores. Now, zoospores is the infective stage of Phytophthora. So you float some susceptible plant material on the water surface, and those zoospores hopefully swim up to the surface, infect that material, and form visible lesions, which you can then plate up onto agar media with the aim of obtaining pure isolates of any Phytophthora species that were present in your soil sample. So, using my soil samples from Kaituna Valley, I carried out this process, but I didn't get any lesions. That's sad, but it raises the question of whether there's truly no Phytophthora molecule in my soil samples, or whether I was just unable to detect it with the method that I applied. And it highlights a key limitation of soil baiting and pathogen testing in general, which is that it's really hard to tell whether a negative result is a true negative or a false negative. There also might be some biases related to the methods that you use for soil baiting. For example, certain species might be more likely to be detected than others, and certain bait species that you use for those leaves might be more susceptible than others. For example, me and Matt, who you'll hear from in a few minutes, did some small-scale trials with water baiting, and we found that Phytophthora doesn't really like pittosporan leaves, but it does really like tea turkey leaves. It's just a bit picky. Additionally, soil baiting is not as sensitive as PCR at low levels of Phytophthora inoculum. So why does it all matter? Well, my QPCR results highlight the potential for exotic forest, which makes up about 8% of New Zealand's land area, to act as a reservoir for Phytophthora. Land use mosaics are widespread across New Zealand and occur in both spatial and temporal senses. In a spatial land use mosaic, there's potential for Phytophthora populations to transfer from land uses such as exotic forest to nearby susceptible land uses. And in a temporal mosaic, there's potential for prior land uses to have Phytophthora-related legacy effects on future land uses. My results also highlight a need for more research into the effect of land use on abundance and diversity of these critically important plant pathogens. That research should include more application, compare more different land uses, and consider other methods like metabarcoding to overcome some of the limitations of the methods that I investigated. So yeah, thank you to Alexa, who's my main supervisor, and Alana and Nat, who are actually part of it. Yeah.
Nicely done. Does anyone have any questions? I'll start with one then. What are the which life stages are harder to extract with the DNA, and are those life stages that are more difficult and tricky? Are they different to the tricky stages in the baiting?
Yes, so oospores are the main life stage that's harder to extract DNA from. They're kind of quite a resistant life stage involved in sexual reproduction. And in terms of the soil baiting, it doesn't matter so much what life stage Phytophthora is in, because you carry out this series of steps. It involves just like leaving your samples to dry for a bit and then moistening them for a bit and then covering them and yeah, some stuff like that. So that process is designed to get them to produce sporangia, which will then release those spores. So it doesn't matter so much what the initial inoculum is, what life stage it's in.
Great. Has anyone else got a question?
Yeah, I've got a question. So I'm wondering, to get around that false negative problem, can you have like a positive control that you have? So you have soil samples that you put Phytophthora in so that you know that should come up, and if you do the same methods, it should see it. Or
yeah, totally. That is like something you could do. I didn't do it, but some people have done that with the kind of very similar methods, and I've found it to work.
Okay,
but
is it a lot more work or?
No, I realistically probably should have done that. It'd be a good idea. It would require having some like Phytophthora to put in the sample.
Yeah,
but no.
Great. Any other questions?
Just building from Nicola's question. you might have mentioned this in the presentation, but the samples that you had a negative response from your soil mapping assay, were they positive, the same soil samples, were they positive in the QPCR?
Yes.
Okay.
Which could be to do with just the level of inoculums. It might just be not enough to be detected with soil data.
How many bait plants did you have?
I used 3 different species. So Pinus radiata, tea toffee, and rhododendron.
Great. All right, we will move on to the next speaker, who is Pearl Ruston from Victoria Uni. So whenever you're ready, Pearl.
Screen share. Cool. Can you guys all hear me and see the perfect? Cool. Tēnā koutou katoa. My name's Pearl Ruston. I'm here today to talk to you about my summer project titled Mapping Myrtle Rust Risk to Maina Tawake. The supervisors of this project were Dr. Julia DeLip from Te Herenga Waka and Dr. Mahachaman Pansy from Manaki Whenua Landcare Research. Maina Tawake, known in Latin as Sigizium maidae, is a nationally endemic obligate wetland species. It is ecologically and culturally important to Aotearoa, with its berries being used by Māori for waiwaka puddings and its bark being used to create dyes for dyeing harakeke. However, 90% of New Zealand wetlands have been lost since European settlement, causing a decline in the Maina Tawake population. Additionally, with the introduction of myrtle rust in 2017, the Maina Tawake population has declined further, reaching a nationally critical state, meaning it is at high risk of extinction. Myrtle rust attacks the new shoots and tips of plants, causing plant dieback. And it's not only Maina Tawake myrtle rust infects, it infects a variety of native and non-native Metaceae species. The idea that myrtle rust infects a variety of different hosts introduces the idea of a pathogen spillover effect, where the main concern would be localized extinction of Maina Tawake. Something seen in Prerecorded frogs due to the cytrid fungus. So in order to combat this, to ensure Maina Tawake doesn't go extinct, the aim of this project was to map Metaceae species across New Zealand to understand myrtle rust infection risk to Maina Tawake. The first steps of this research was deciding the 20 species I wanted to focus on. I chose 11 native and 9 non-native Metaceae based on their level of susceptibility and abundance in Aotearoa. This was backed up by researchers at Department of Conservation and Manaki Whenua. The next step involved collating as many records as I could of the 20 species by talking to New Zealand Herbaria, online Herbaria, PhD students' records, and the National Vegetation Survey. I compiled the data and input it through QGIS to create distribution maps. The first map is this distribution of Sigizium maidae, of which Sigizium maidae is located in the North Island, lowlands of the North Island and the top of the South Island, as you can see here. And here is the distribution of native Metaceae of Aotearoa, and here in blue, distribution of the non-native Metaceae. Just by the sheer abundance of native Metaceae, the density and just wide distribution of native Metaceae, native Metaceae poses a greater risk to Maina Tawake in comparison to non-native Metaceae. But the sheer difference between the two numbers is understandable, with native Metaceae having earliest record being from 1877. With that in mind, non-native Metaceae are expected to increase over time and have increased. Here you can see the non-native distribution pre-1991 and here post-1991. There's an increase in non-native Metaceae, and the spatial density in more urban areas has also increased. My first thought was potentially a spatial bias here, as humans are more likely to observe things in their backyard than in rural areas. But non-native Metaceae such as Sigisium smithi and Sigisium australe are becoming known as common lily pilly, being commonly found in people's backyards and sold in nurseries as they make for excellent hedging. Thus, this trend is acceptable and makes sense. Yeah. So the distribution of all these maps have allowed us to understand how infection risk changes by density through space, depending on Metaceae distribution, and over time as Metaceae distribution changes, therefore allowing us to reach the aim of this hypothesis, which was to gather some understanding of myrtle rust infection risk to Māori Tawake across Aotearoa. Cool. I would like to just kind of mention where the data will be going in the future. We'll be heading off to a postdoc in Dr. Julie Glep's lab, who will use this data with other data to get a better understanding of myrtle rust infection risk to Māori Tawake restoration. Yeah, so I'm really grateful to have been part of this project and I've learned a lot along the way. I can't wait to see what happens in this space. And thank you to all these people and Bioprotection Aotearoa for this amazing opportunity. Ngā mihi. Thanks, that's me. Happy to answer any questions you may have. Cheers.
It's wonderful. Thank you, Pearl. Has anybody got any questions? Pearl? I guess for me, I'm just hoping this is a really good reason that we can get rid of some of these hideous invasive weeds that are widespread in bush patches around Auckland. So maybe this is another good reason to get rid of them.
Yeah.
Anyone got any questions for Pearl?
Nothing from down here.
Pearl, had you used GIS before, or was this a new skill for you?
A new skill for me, so I definitely think it's going to come in handy with future research, but it definitely took a while, lots of in the computer lab figuring it all out, but enjoyed it.
Yeah, and an important one for jobs too, so well done you for picking up some new skills there. Thank you very much. So we're going to move on to Tara Curtis now, who I think is in person there. She's from University of Canterbury.
Well, we just got a question that's just popped up.
Oh, great. Let's do that.
Yeah.
Oh, okay. What are the main exotic myrtle species that people seem to be planting in their gardens?
That would be Sigisium australe and Sigisium smithi, just kind of common lily pilly, just make for beautiful hedging. So people want them. My auntie even mentioned that's why she plants them, because she quite likes them. And I'm like, no, but yeah. Thanks.
Yeah, good answer, Pearl. We need more of you. And certainly a species that my summer students in Diana have been picking up in our bush patch plots. So we want to get rid of those. Okay, so Tara, ready to go now? She's in person.
She's here in person. I'm just
Yay. Here we go. So Tara's from the University of Canterbury. And whenever slides come up and you're ready to go, Tara.
Hey. So great. I worked with Claudia Milsrumler's group under, sorry if I messed it up, under Amanda Board, and I was helping her with her PhD project, which focused on oleosin. And this is one of the ways that we wanted to examine oleosins, how to characterize oleosin. So some background. Triglycerides are an important fuel in seed germination, and they're very insoluble though. So they need to be protected, and it's through this phospholipid layer that they are protected, which has many proteins, and the most abundant is oleosin. So oleosin structure consists of this N-terminal, then this long hydrophobic domain with a proline knot right at the end, and the C-terminal. And the C-terminal and the N-terminal typically sit on top of the phospholipid layer. This is meant to supposedly play a role in stopping the oil bodies from interacting with each other, like clumping together.
And this long hydrophobic domain is about 70 residues long usually, which is, we wanted to investigate how important this is. So the way the oil bodies are formed is the ribosome mRNA complex is transported to the endoplasmic reticulum, where it is translated, and the main method thought to how these oil bodies are produced is just as more triglycerides and proteins are synthesized, it'll just eventually bud off. And you can see, so how we want to investigate how important the length was we wanted to make a series of truncations and see if this length was really integral to the structure and function. So the first truncation we made was a really large truncation because it's usually about 70 residues, the hydrophobic domain. So we truncated it to 36. And then from then on, we did a series of reducing 4 residues at a time until the last one, which was only 3 residues because the proline knot was the 13 residues, and that was essential to synthesis. So we wanted to keep that, helpful. And then the way we investigated what effect this had on the oil bodies was we infiltrated it into the Nicotiana plant in agrobacterium. And then we were able to take these images of the oil bodies inside the plant. So this is the wild type, which was our control, and you can see it's a bit overexpressed in the first one. But so we were able to reduce the expression, and you could see it create these really bright circles around the nucleus, which is where the ER is located, and so that's where the oil bodies are being synthesized. So that's what we wanted to look for in the truncations. So these are our results, and for the first truncation was the 36. Here again, you can see these beautiful bright circles around the nucleus that show the oil bodies are still being produced in the ER, and then these little circles floating around, those are also oil bodies just free doing their own thing. But from 32 onwards, we saw that instead of circling the nucleus, they were like really just filled the whole nucleus. They looked like little eyes. So we saw these bright circles, which kind of tell us that they're probably soluble now. So they've lost their function, the whole point of what they're meant to do. And it was like that for the whole series until 13. So what we learned from this was that the length of the hydrophobic domain is really, really important actually to the structure and function. And when this is reduced too much, it no longer functions. And so that's it. Sorry. And I'd like to thank Claudia and Amanda and obviously Bioprotection for funding my summer. Thank you.
That was awesome. Has anyone got any questions for Tara? Don't make me try and ask a question about something I don't understand. I'll ask you, did you learn new techniques, or were those some of the techniques you already learned at undergrad or postgrad?
Everything was new.
Cool.
I feel like I did nothing in undergrad. I got to finally actually do some techniques. It was really good.
That's what we like to hear.
Patients as well as the microphone.
Amanda did the truncations. I came in a bit later. I was just helping put everything in. Did all the application. Yeah. Everything was ready to go.
Any other questions for Tara?
We've got an online question here, I think.
Okay. Oh, what was a great question, Irene? What was one of your highlights during that process?
Oh, it's a good one. I really liked infiltrating the plants actually, because I give them words of affirmation. I hope that they grow better, and I think it worked. That was my favorite part.
That's awesome. Great. Thank you very much. So now we're going to move on to Max Oxenham from Lincoln Uni, who is online with us. So whenever you're ready to share your screen, Matt,
Can you guys hear me all good?
Yep, perfect.
Cool. So we'll just cruise across here and hopefully that'll work. Awesome. So hi guys, my name is Matt Oxenham. I'm at Lincoln and I'm getting very close to finishing my Bachelor of Science, Environmental Science and Environmental Management as well. So over the summer I got to work with Alana, Finn and Leodin. And as we'll see, me and Leodin's presentations have a bit of a crossover. So the general topic that I was working on was understanding the role of microbes in resilient soil ecosystems. And my little title I've given it is Water Mushrooms and Tree Fungi. And the reason I've called it that is because there seemed to be a bit of a trend over the summer of me trying to explain to my friends and family what I was doing, them not really understanding and me just having to say I'm doing water mushrooms and tree fungi, and that seemed to work. Okay. So the structure of my summer was a bit all over the place. I didn't have too much direction coming in. So instead of having my own specific research aim, I kind of just worked alongside Finn and Alana on their projects and helped them out however I could, and learned some of the methods that they were doing. So Finn's project involved characterizing AMF fungi found in native and agricultural ecosystems. And Alana's project was addressing knowledge gaps. Go back to that slide. And soil resiliency by looking at potential for Phytophthora invasion up in Te Kaha. Cool. So the first bit of work I did here, I've got a video for you guys, was just mucking around with some AMF spore isolations. So that process was pretty much just sieving soil, using a sucrose solution and then putting them on a petri dish or putting them on filter paper. And I was trying to understand like the different relative abundance of these AMF fungi across the different land use types, which literally meant counting these spores one at a time to try and understand the differences. And the result of that was a bit of frustration occasionally and the spore murders you're seeing on this screen right now. Cool. So I used that method a bit and I was trying to understand abundance, but I think there was a series of inaccuracies, which meant that this method is a little bit outdated for doing so, but it is still really useful for doing sort of morphological analysis of a few different spores. So that was my work with Finn. And next, I did some work with Alana. So we were doing Phytophthora baiting. We did a trial, the main goals of which were to understand the effects of different land uses on the Phytophthora that you end up with in this waterway. We went up to Kaituna Valley and did some river baiting. And also they just served as an opportunity for Alana to try out some of her methods before we went and did them up in Te Kaha over there. So river baiting for Phytophthora involves setting up these little mesh bags, which is what you can see on the top left, with your different baits, suspending them in the water column, leaving them for a few days, and then hopefully you end up with some lovely lesions like you see on the bottom left here. So yeah, we set up a little trial. Eventually you come back, take cuttings of those lesions and put them on some Phytophthora selective agar, and they'll start growing. And then from this point, I started trying to identify them. So as you can see on the left, you get one of your first features, which is a different growth rate on this Phytophthora that grew on pine. And then also some of them start to present these lovely growth patterns like you see on this middle photo here. This is a nice like rosette pattern. And then the one on the right is more typical of what we sort of found. After this, I started doing a bit of microscopy to identify some other features. And this one here, luckily, showed us some lovely sporangium. And then those little blue dots inside the sporangium there, those are the zoospores. So I started just going through and measuring different features like your hyphal widths, different features in the sporangium and other spores, and trying to identify them. And finally, just to tie things in, this is what I believe to be the Phytophthora presenting themselves in their true form, which is a nice little water mushroom there on the screen. Cool. So thank you very much for all your guys' help over the summer. It was a good learning process. I remember my first day in the lab, Leodin had to show me how to use a pipette. So I came in with virtually no experience pretty much. And it was a really good experience to put some of that book knowledge and sort of soil microbial ecology, into some actual practical skills. So cheers.
Awesome. Thank you very much, Matt. Anyone got any questions for Matt? So Irene wants to know what's a new skill you picked up over the summer.
As I said, I didn't really know how to do anything in the lab. I did some biology papers in first year, but they got cut off nicely by COVID, so I got about one lab. So everything down to making agar, culturing, subculturing, microscope work, it was pretty much all fresh to me.
Yeah. And nice combination of getting out in the field as well as in the lab by the sounds of it.
Yeah, exactly. It was amazing to go up and do some work over in Te Kaha as well. And yeah, dip my toes in as much as possible, which is a really good experience.
Yeah. And perfect timing to dip your toes in at the beach up there. It sounds awful. And can I ask, you were baiting different leaves. How did you and Alana make the choice about what leaf species you were using there?
Yeah, so you want to pick leaves that are somewhat thick and waxy is what one of another resource we saw told us. You obviously don't want something that's just going to completely decompose over that time. And then I think it's also just a matter of like trial and error. There sort of seems to be ones that are more effective, like people mostly use rhododendron. That seems to be a really good one. We found that Titoki was really good. And then for Alana's baiting up north, she used corn and kiwi fruit leaves because that's what's going to be investigating up there.
Yeah, makes sense. Cool. Nicola Day is asking, how many species of AMF spores did you see?
Oh, I saw a lot of spores. I didn't really get around to like identifying the different species of spores there.
Just lots of different ones.
Just lots of different ones. And I had guides which sort of outlined different spores of other types. So these, like some of these spores here, like these really dark ones that you can see, aren't AMF spores. They tend to be the lighter colors and the brown colors and that kind of thing.
Cool. Awesome. Thank you very much, Matt.
Cheers, guys.
And last but definitely not least is Alice Spears, who's at the University of Auckland. So she's going to be finishing us off with another weed talk.
Hello, everyone. I will just share my screen. Hang on a minute. What are you guys seeing at the moment?
We're just seeing your desktop with it sitting on there. So I think you need to go to the bottom right of your PowerPoint. See that little one that looks like a screen down the bottom by the Zoom button on the left? Sorry,
No, we might have it.
Cool.
Can you see that?
Yeah, perfect.
Okay. So, okay. So to everyone, my name is Ella. I'm from the University of Auckland. And this summer, I stepped well and truly out of my animal comfort zone and worked on a project that investigated the impact of multiple weeds on restoration planting. So I was specifically looking at how bird dispersal can influence what's growing in the understory below three main species: tree privet up here, I don't know if you guys can see my mouse, woolly nightshade at the bottom right, and then brush wattle on the bottom left here. So as we can see, tree privet and woolly nightshade both have their seeds encompassed in these big fleshy fruits, which entices birds such as blackbirds and silverise to come and eat them. But brush wattle here is slightly different in that it uses explosive dispersal to spread its seeds around. This is where the seeds are forced out from the plant by the pressure of the seed pods drying out. So because these birds like eating these fleshy fruits in the nature of brush wattle's dispersal mechanism, I hypothesized that there will be a difference between the amount of bird dispersed species underneath fleshy fruited tree privet and woolly nightshade compared to brush wattle. And this again is just because blackbirds and silverise don't really have any incentive to come to brush wattle if there's no fruit to entice them over. So I recorded all of the weed species within a 4 square metre circumference with one of these 3 subject species in the middle. I also measured the height of the central species, and these observations were taken from around Greater Auckland between December and February. And here is just a little map of my site. The diagram on the right here shows the design of the seeding plots we were sampling. So if we look at this graph here, the red bars represent tree privet and woolly nightshade together as they are both bird dispersed. This is showing the percentage of weed species seedlings and plots under central weed species on the y-axis up here, and then whether they are dispersed by birds or other mechanisms on the X down here. So as we know, again, I'm predicting a difference between bird dispersed species underneath these 2 groups of weeds. But this is science. We can see that there are in fact more bird dispersed species underneath brush wattle in blue than under the fleshy fruited species. Not shown is a table of the most commonly found species per dispersal type. Interestingly, woolly nightshade seedlings were prevalent in the understory for bird dispersal. In terms of reasons for why we might be seeing more bird dispersed species under brush wattle, woolly nightshade is actually allelopathic, which means it has strong below ground chemicals that inhibit the growth of its neighbours. The representation of species in plots with woolly nightshade may therefore not be as representative of richness or abundance as we would otherwise see in the absence of the species. Alternatively, there's been some work done that suggested birds may preferentially perch from higher vegetation. In this case, what we're seeing may be that birds are actually going to woolly nightshade and tree privet to eat the fruit, but then leaving and going to higher vegetation such as brush wattle to excrete these or poo these out. This would explain the high abundance of bird dispersed species under brush wattle then. And I did not take this perch tree effect into consideration when I was formulating my hypothesis, but that seems like a pretty feasible explanation to explain why there is no difference in bird dispersed species under tree privet and woolly nightshade when compared to brush wattle. And on the right here, we just have this little diagram of brush wattle as the central species in a plot. It's presumably the highest plant around, and so we have this blackbird coming in to perch on the tree, poo, and then deposit these fleshy fruited species such as woolly nightshade that it has digested previously. So in conclusion, I did not find any evidence that there is a difference in weed species under tree privet and woolly nightshade when compared to brush wattle. But this has been the most incredible research experience. I've learned so much about these species and weed dynamics in general, but also about the nature of science itself. It's definitely not the linear path that I initially thought it was. Thank you so much to Margaret and Diana for all your help and support, and a huge thank you to Bioprotection Aotearoa for the welcoming and encouraging community this summer. It's been so nice to feel part of a scientific community, and I've really valued my time here. Thank you.
Thank you, Ella. Has anyone got any questions for Ella? Maybe your own supervisor should ask you a question, Ella. Uh-oh. So are you going to, after all of these things that have happened, you still keen to do masters?
Yeah, I think so. Just tip my toes in now. Got to see what it's all about.
So I should just make sure you sign that dotted line first then.
Yeah, I'll get there.
Okay, I think everyone's running out of steam a bit. Any last questions?
Maybe I was just wondering if you saw any specific birds going into those bushes while doing the experiment or the survey.
Sorry, are you able to repeat that? It's just a little bit
So
fuzzy.
the question was, did you see any birds come into these plants while you were out there doing the survey?
No, so when we started going out, I thought that I would take bird counts or like 5-minute bird counts or observations of birds around. But when I thought about it, I thought that's probably not a very representative way of doing it, as what we're seeing is going to be, just what's there at that immediate point in time. And we were going to heaps different places at different times of the day, different, points in the season and everything. So I thought that probably wasn't going to be a very reliable proxy of what is there. So no, we didn't take into consideration what was actually there.
Great, thanks, Ella. So Ella is the last speaker for today. And before I kind of open the floor for more questions or comments, I think it's worth just everybody having another round of applause for our wonderful students. So thank you very much for your hard work and enthusiasm, because, you know, these old people tend to get a bit cynical sometimes. So we like when these new enthusiastic students come on board. And a particular thanks to all the supervisors and master students and PhD students and lab groups that supported all of our students over the summer, because that's a really important part of creating a culture that our students will enjoy. So thank you all for your manaakitanga towards our summer students. So I'm going to open the floor for any last questions or comments, and then I'm going to hand it back to Zohar to close. Anyone else got any comments or questions? Diana?
I was going to ask, thank you. I was going to ask Ella, did you look at how many sites we actually had for the brush wattle? Because I think I remember we only had like 2 or 3 brush wattle plots to compare at that point.
Yeah, there was a bit of a species skew, I think, in our data. So in the height measurements that I did look at, it was the median height and not the average, because brush wattle was doing some crazy things that weren't really, you know, showing what I wanted it to. But yeah, definitely we had a lot more tree privet and woolly nightshade than brush wattle in our observations for sure.
Anyone have any other questions for any other students that have been stewing along in your head? I'm going to pass back to Zohar then.
Awesome. Well, thanks so much. I just wanted to reiterate some of the comments that are coming through the chat. It was awesome to hear everyone's presentations and all the hard work that you guys have done and put in over the summer. And it's awesome to see our wider BA whānau like coming in to awhi all our presenters today. So thank you very much for everyone being here and involved over the summer. And, you know, once you're part of the BO whānau, you're always part of the BO whānau. So thank you. And we can look forward to continue working with you all going forward. So with that, we will close for the day. We will put the presentation on YouTube and on our website for everyone to look at who was not able to make it today. And thank you very much for being here. Enjoy the rest of your day. Oh, sorry. Biggest thing. Thank you, Margaret Stanley, for coming on. Sorry. Oh my gosh. Biggest faux pas there for stepping in and being our wonderful host. You have set the bar high, continued the expectation that Phil Hume is left behind from last year. And watch out who's going to be next year. Any volunteers?
I feel like Phil wouldn't have forgotten to show the video.
Well, there was no video last year.
Thanks all.
Thank you. Cool. The people that were there. Oh, hiding behind the screen.